Hello and good evening. This is Donnie Rooney, and I am presenting over the protein DNA helicase. So, to start off, just an introduction on viruses in general. So, protein functions are vast. I mean, you've got stuff anywhere that has to do with DNA replication, to membrane structure, to just about any job or task you can think of. I mean, even energy synthesis within ATP. And with protein structure, you've got four levels of protein structure. You got primary, which is going to be the amino acid sequence; secondary, which is going to be those chains of amino acids; tertiary, which is going to be the foldings, alpha helices, beta pleated sheets, etc., of how those polypeptide chains fold onto each other; and quaternary is going to be multiple of these polypeptide chains joined together to make one full protein. And one thing to remember is that structure and function is connected. Without the correct structure, if there's any kind of mistake or weakness or broken chain, to name an analogy, then the whole protein could break down and its function would be incorrect. So down here is just a quick image on the structure of proteins. I mean, if you'll see over here, these are pointing at individual amino acids, and the whole thing is going to be your amino acid chain. And what this doesn't show is that it can fold over and do different stuff, alpha helices, beta pleated sheets, etc. And when you get multiple of these formed together, or even just one, you can have a full protein. So one thing to point out is that not all proteins have a quaternary structure. Some of them stop at tertiary, some of the less complex ones. Which is not the case for DNA helicase. And you'll see that this picture is it's insignificant as to what it is, just the picture showing, and that's where it came from. So DNA helicase, what does it do? Its function is that it unwinds the DNA double helix. Down here in this picture, that's basically what it does. You've got the two strands of DNA, and ahead of it they're still coiled up, and as it goes through, it cuts the hydrogen bonds in between and allows the other proteins involved in DNA replication to create the new strand attached onto these two separate strands that are being cut apart. So it's semi-conservative. And DNA helicase is found. It can be found pretty much anywhere that there's DNA, because the way that DNA replicates that we've seen today is in this form. I mean, I'll show later a ribbon model of E. coli's DNA helicase, and it's actually really similar to humans, because no matter what the organism is, if it has DNA, if it's got the double helix, it tends to form this spiral form like it has, and we'll cut it in half like that, no matter what the organism is. So, the structure of DNA helicase. I didn't find any specific amino acid sequence, but one can assume there's going to be just a ton of amino acids and a bunch of different chains formed together. So the secondary structure is going to be how those amino acids form these chains. And a tertiary structure, each one of these chains is going to fold in its own way, and then the quaternary is when all these chains are going to come together. Now, over here in the picture is really where you can sort of get a sense of what I mean when I'm saying all this. So one can assume that each of the different colors on here, you've got yellow, green, blue, all kinds of different stuff, and even some stuff in the background there. One can assume that each one of those colored strands is going to be a different polypeptide chain. So, there's a bunch of different chains in there, and DNA helicase wouldn't be complete if it were missing any of those chains. So, you can see definitely plenty of the alpha helices, and then you can see what could be construed as some beta pleated sheets that have circled around themselves into a cylinder. And this is actually a picture of a DNA helicase of E. coli, not of the human DNA helicase. In my paper, there is a picture of DNA helicase, and it's actually really similar to this. Now, DNA helicase, genetic information and diseases. So, first off, the gene sequence. Now, honestly, I don't know what this stuff means, but this is your sequencing of the genes of DNA helicase, and this is of, I believe, DNA helicase B of humans. So I'm sure that they've got just classes and that focus on this specifically. So the importance is not so much what it all means. The importance is that it has been sequenced. There has been plenty of research done into it, and I mean nowadays you can go and get an entire human genome sequenced. I believe it's for like $10,000, and it'll take maybe a few days or a week, maybe a month to get the information back to you. So this would most likely be relatively simple compared to that. And of course, there's going to be some diseases associated with problems of DNA helicase. The diseases Bloom syndrome and Werner's syndrome. What Bloom syndrome is basically the symptoms that you see are going to be stunted growth and an increase in cancer risk, especially when exposed to direct sunlight. And this one's actually really rare. There's a handful of cases ever recorded. And then Werner's syndrome is going to be sort of premature aging. Those who suffer from this see symptoms of, just the things that you'd see with older people, and the symptoms that come with aging, and there's some stunted growth issues, and it's more common. I believe the number is like 1 in 200,000. At the end of this, I have the website I went to. It's got all that information on it. So, in conclusion, with DNA helicase, like many proteins, it's very complex. Simply put, it unwinds a double helix for DNA replication, and it can be found just about anywhere that there's DNA. Of course, it'd be different slightly, most likely, between each organism, but overall the structure is going to be pretty close to the same because the functions are going to be pretty close to the same. And this protein does show all 4 levels of protein structure. It has been sequenced genomically, and there are a couple of diseases that have been associated with it. And one thing I like to point out too, that Werner's disease is actually not a problem of DNA helicase itself. It's actually a problem of a gene that's associated with it's a gene that the mistake in it causes a free-floating sort of unresponsive protein, and this protein can interfere with many of the basic cell tasks. And so DNA helicase is currently being theorized as one of the proteins that is affected by this protein that's not needed. And that is my presentation of DNA helicase, and here I'll stay for just an extra minute or two, or 20 seconds or so, just so you can get a good look at all of these sources that I used. Thank you. Good night.